That means in, you know that how a single cells, that means producing that of two cells like this, one parent cell, one mother cell producing two daughter cells. But today what you will have to discuss about that of that is meiosis. Okay, this is meiosis, and in that meiosis you will have to know that how it is generally found in that of sexual organisms and in the sex cells, and it is generally found in the sex cells, and it is generally what it does that in haploid cells, in haploid, that is in haploid cells, that means it maintains the haploid, but after fertilization the diploid is maintained, and it is generally okay. That means you see that that means it is generally found in that of diploid cells, but in that sense that it is always that chromosome numbers always become half. So the half chromosome numbers and half chromosome number of male to that of half chromosome number of females. So in such case of a haploidy of that of male gamete to that of haploidy of female gamete becomes diploidy after fertilization. So that you can see here that in meiosis, how that means haploid in that of chromosome is maintained, and after the fertilization, how the diploid is restored. And in this also you can know about that of four cells. That means in that of mitosis, that one mother cells producing that of two cells, but here we can get that of meiosis, that producing to that of four daughter cells. But in mitosis there is a one cell division, but here in meiosis you can have to find that of two cell divisions. Okay, two cell division. There are two events of cell division in meiosis, that is meiosis 1 and meiosis 2. And meiosis, in between meiosis 1 and meiosis 2, there is students, there is a gap. That gap is called interkinesis. There is a resting phase. That resting phase is called interkinesis, but it is for that of for a momentary. It is a momentary affairs, and it is in between that of meiosis 1 and meiosis 2. There is a phase. That phase is called interkinesis, but it is just a transitory phase, and it is just for the few seconds. Okay, after that meiosis 2 will start, and meiosis 1 is called that of reduction division, because it will reduce the chromosome number. Suppose chromosome numbers in human being is that of 46 number of chromosomes. Meiosis function is to reduce it to that of 23. Okay, but in that of meiosis 2, it is equal to that of mitosis. So mitosis and meiosis both some way similar. Owing to that region, it is called that of equational division. And in that of meiosis 1 and meiosis 2, you will have to find that four subphases, that is prophase, metaphase, anaphase, and telophase. For that of meiosis 1, it is called that is prophase 1, metaphase 1, anaphase 1, telophase 1. For that of meiosis 2 also you can know about that of that is prophase 2, that is metaphase 2, anaphase 2, and that is telophase 2. In such way you can have to discuss today that of meiosis. Okay students, now you ready for that of meiosis. Now you see that is meiosis. Okay, meiosis. You see that the term meiosis was first used by Fermi and Murray. By Fermi, that the term meiosis was introduced for the first time by Fermi and Murray. In 1905. Okay, in 1905. that is partition of Bengal. In that, in Indian history, that 1905 is famous for that of partition of Bengal. Okay, so that of that is Farme and Murray, that means proposed that of meiosis, introduced the term meiosis for the first times, and elaborately discuss about that of that purpose of that of meiosis. Okay, that objective of that of meiosis. So who introduced? That is Farme and Murray. In which year? In 1905. Okay, 1905. And that means that meiosis involves that of two sequential. That meiosis involves two sequential, that is two sequential nuclear division. That is nucleus division, but one event of one time of that is DNA synthesis. Okay, DNA synthesis. That is meiosis involved two sequential nucleus division. That means there will be a two cell division. That is, one is that of that is meiosis 1, another one is that of meiosis, that is meiosis 2. Okay, meiosis 2. Meiosis 1, that is number of chromosomes will be half. So that it is called that of reduction division. But it is some way equivalent to that of mitosis. Going to that region, it is called equational division. Okay, division. But DNA synthesis will occur only in that of interphase before the starting of meiosis. Like that of mitosis, in meiosis there is also an interphase stage. Okay, interphase stage. But basic difference is that in mitosis that S phase is some way shorter, but in meiosis S phase is some way longer, and that G2 phase in interphase of that of mitosis is shorter, but here in that of meiosis the G2 phase, gap 2 phase is very, that means it takes very little times. That is, but in some species, in some cases also, that gap 2 phase is also absent. Okay, it is shorter. It is the shortest. That means gap 2 phase is shortest compared to that of the gap 2 phase before mitosis. But in meiosis, it is shortest, and in some cell division, that gap 2 phase also absent. Okay, so there is a interphase in before that of meiosis 1. Before meiosis 1, there is an interphase. Okay, interphase. That is an interphase, but before meiosis 2, there is no interphase. That concept should be clear. Okay, before meiosis 2, there is no interphase. Before meiosis 1, that is a interphase stage. And that interphase is same also. Okay, that interphase also same, like that of G1, S and G2 phase. But that G1 S phase, is longer comparatively to that of S phase of mitosis. Okay, but gap 2 phase is the shortest phase, and in some organisms, that means this gap 2 phase is absent. After synthesis, directly meiosis 1, that is prophase 1 starts. Okay, that point we should have to remember. And but as meiosis, after meiosis 1, meiosis 2 starts, so there is no interphase. before meiosis 2. Only interphase found before meiosis 1. Interphase found before meiosis 1, not before meiosis 2. But in between that of meiosis 1 and meiosis 2, there is a there is a interval. That is, there is an interval. That interval is called that of interkinesis. Okay, that is called interkinesis. Okay, that is called interkinesis. And after that, meiosis 1, same, that is karyokinesis. After karyokinesis, in meiosis 1 also same, that is like mitosis, karyokinesis. Karyokinesis involve that of that is, that prophase, metaphase, anaphase, telophase, like that. And that also, so meiosis 2 also same, that of prophase 2, that is metaphase 2, anaphase 2, telophase 2. And but that of meiosis 1, that is meiosis 1, in meiosis 1, that you can have to find that nucleus will be divided into two parts. Again, also in meiosis 2, also nucleus will be divided into two parts. Okay, two halves. That means nucleus will be divided here, nucleus will be divided here also. But DNA synthesis is same, that is in S phase. That is DNA synthesis. Okay, DNA synthesis will occur only that of S phase. S phase. And that meiosis 1 and meiosis 2, and meiosis 1, especially for meiosis, that is meiosis 1, that is a reduction division, and you will have to know that how the chromosome numbers become half. And in that case of the meiosis 2, you will see that how that actually that meiosis 1 completes all the functions of the meiosis and fulfill all the objective of the meiosis 1. But meiosis 2, what is the necessity of meiosis 2? I can also tell you that what is the necessity of meiosis 2. Meiosis 1 completes all the functions of all the objective, fulfilling all the objective of meiosis. But why there is a necessity of meiosis 2? I'll also explain later on this chapter, that is portions that I am teaching now. You can have to know it. But first of all, you will have to know about that of meiosis 1. Okay, meiosis 1. So what you have know up to that, meiosis is that of that ensure the production of a haploid phage. Okay, haploid phage. In the life cycle of a haploid phage, it ensure the haploid phage. That haploid phase of that of a haploid phase of the sexually producing organism. Haploid phase of the sexually reproducing organisms, and meiosis generally found in that of gonads. Okay, it is found in that of sex cells. Sex cells, that is called gonads. Okay, it is found in gonads. Gonads means it is found both in, that is it is found in testis as well as also in that of ovary. Okay, so testis and ovary, that is gonads, and also in that of spore mother cells, that is gamete mother cells. In gonads means where, that is gamete mother cells, and that of spore mother cells. In gamete mother cell and spore mother cells of gonads, that meiosis is generally found. Okay, it's generally found. And in that gamete mother cells, it will have to produce, that is haploid male gamete and that of a female gamete. Okay, sperms and egg will be produced, that is haploid, that is N. And spore mother cells also produce that haploid spores. Okay, so meiosis is generally found in gonads, in sex cells, and it is in that is gamete mother cells or that of spore mother cells. Okay, and that the term meiosis was introduced by the Harme and Miro for the first time in 1905, and that means haploid converts into diploid at the time of, after that of fertilization. Okay, fertilization. So N and N, that N combines together when after fertilization it will be 2N. So that diploid is restored. In meiosis also, haploid meiosis, that means chromosome numbers half. If it will not possible, if there is no meiosis, students, you see that. That means suppose a diploid organism 2N, and if there is no that of there is no meiosis, then this 2N, that will be 4N, then it will be 18 like this. So the chromosome numbers become increased. So it will, that means it is biologically it is not possible, and biologically you can have to bring the stability in the species. So in order to maintain the stability in the species and to have that of equal chromosome numbers in species, that is meiosis is absolutely necessary. Okay, so that's about that of introductory part of that of meiosis. Okay, meiosis. Then we'll have to go to that of students, that of that is meiosis 1. Okay, meiosis 1. Meiosis 1. So if you have to make that of you see that here is that of I'm dividing it. Here you see that is gap 1, that is S, and that is gap 2. Here you can have to see that here is that of okay, and in between you will have to get, here is that of meiosis 1, that is meiosis first. Here is that of meiosis first. Here we will have to find out that of interkinesis, that is interkinesis, that is interval period. Then you will have to that meiosis 2. Okay, here we will have to, that is karyokinesis. Here is that of after karyokinesis, that is cytokinesis. So here is that of cytokinesis. So then you'll have to come to that of that is gap 1, gap, that is, and gap 2. Here also students, here also after meiosis 1 also there is also cytokinesis. Here also cytokinesis. Okay, cytokinesis. So G1 is G2, and a DNA is synthesized only in that of S, that is DNA synthesis of DNA. And the same function that of what in mitosis, same is also in that of meiosis. In that of that is organelles, that is organelles, how organelles are being different organelles are synthesized in that period. Our RNA, that is synthesis, protein synthesis. Here also that of centrioles. Okay, that is centriole, duplication of centriole. Here also you can have to protein synthesis, RNA synthesis, and doubling of that of that is shell organelles you can have to find here. Okay, and here meiosis 1, then cytokinesis, then interphase, then meiosis 2, then cytokinesis, then gap 1. Like this, that is that of life cycle of that of meiosis. You can have to know about that of meiosis. Then we'll have to know about that of meiosis 1. Okay, meiosis 1. You see that is meiosis 1. That is meiosis 1. You have to know about that in meiosis 1. In meiosis 1, that has been divided into 4 phases. Okay, meiosis 1. That is first one is phase 1. That is then metaphase 1. Then students you have to anaphase 1. Then students that is telophase. That is telophase 1. Okay, that prophase is that of longest phase. That is, this phase is the longest phase, and it takes the 90%. That is 90%. It is nearing to the 90%. 90%. It is the longest phase of meiosis 1, and it covers to that of 90% times. Okay, so prophase 1. So as it takes the maximum time, it has been divided into 5 subphases. It has been divided into 5 subphases. So prophase 1 is divided into 5 subphases. Okay, 5 subphases. Which are these 5 subphases? That is leptotin. Okay, that students, that is leptotin. Okay, then zygotin, then pachytin. Okay, pachytin, then diplotin. Okay, diplotin. Then carry, that is cytokinesis. That is the last phase, students, is that of Dikinesis, sorry, dikinesis, dikinesis, kinesis. Okay. So meiosis one has been divided into 4 phases: prophase, metaphase, anaphase, telophase. Not difficult for you to remember, as it is also you know, and you will fully acquainted with that of in mitosis. So same thing, but one as it covers the meiosis one. So in prophase one, metaphase one, anaphase one, telophase one. Okay, and the same thing is that prophase one as is the longest phase, that is the longest phase. That means it has been divided into 5 subphases. 5 subphases, okay? 5 subphases. Which are these 5 subphases? Leptotin, zygotin, pachytin, diplotin, and dikinesis. You have to remember in a sequence, but generally in mid questions, there is are in the following subphases of prophase 1 of meiosis 1 in a correct sequence. Or which is the correct sequence? You have to remember that one. That is leptotin, zygotin, pachytin, diplotin, and dikinesis. Okay. So that's meiosis 1's first stage. That is prophase 1. It is the lengthiest one. It's the longest one. So it has been divided. Students, you have divided for better study, for better understanding. Okay, cells have not divided these things. You are human beings. We have divided to gain the knowledge, to know at which time, that means which period generally events are occurs. You see that. That means in mitosis generally take 1 hour or certain minutes for that is for cell divisions. But here in meiosis, that it is take not that is minute or that of hour. It takes some days. Okay, it takes some days. So meiosis takes some days for completing the process of cell division. Whereas in mitosis, it is just that is 24 hours, that is, or that of 90 minutes, or that of 20 minutes. Also, you have known that a bacterial cell, of that of bacterial cells, in that of that is yeast cells, that is 90 minutes. You know about that. So I am not repeating. But here meiosis takes the maximum time. So it is for that of some days also. Okay. So that subphase are leptotin, zygotin, pachytin, diplotin, and dikinesis. First of all, we will have to read that prophase 1. Prophase 1 is that of longest phase. It takes that of 90% of the time of meiosis 1, and it has been divided into 5 subphases, students. Into 5 subphases based on chromosomal behavior. Subphases, 5 subphases on the basis of chromosomal behavior. That is behavior. On the basis of chromosomal behavior, that prophase 1 has been divided into that of 5 subphases. Now we'll have to discuss about that of leptotin. Then we'll move to zygotin, then pachytin, then diplotin, then dikinesis. Okay, I will start with that of that is leptotin. Okay, students. Now we'll have to discuss about that of leptotin. So, can you students tell me about that? That is 5 subphases of leptotin in a sequence. Can you tell, students? Yes, tell.
Sir, 5 subphages of prophase, right?
5 subphages of that of prophase 1. Okay, prophase 1. 5 subphages of prophase 1.
Sir,
First one.
Sir, leptotin, zygotin, pachytin.
No, pachytin.
Sir, leptotin, zygotin, pachytin, diplotin, and dikinesis.
Dikinesis. First one is, I'm just asking you tell. First one is
Leptotin.
Second one is
Zygotin.
Third one is
Pachytin.
Fourth one is
Diplotin.
Fifth one is
Dikinesis.
Okay. We'll have to know about that of these 5 subphases. You know that 5 subphages means what events occur in that prophase 1, you can have to easily know. Okay. For that of on the basis of chromosomal behavior, that means this prophase 1 has been categorized into 5 subphases. First, you will have to know about that of leptotin. Okay, leptotin, students. That means I am just telling you, I am drawing, and I am drawing. You also draw with me. Practice for your practice. That means how many times you will have to write. For how many times you will have to practice, it is better for you. Okay? Because you can have to gain the first-hand knowledge. You see that I am just drawing. You also draw with me. That is leptotin. I am writing here, students. That is leptotin. Okay, leptotin. That is the first subphase of that of meiosis. That is prophase 1. So that in this shell, you see that this is the cell, and in this shell, you see that nuclear membrane. These are the nuclear membranes. Okay, these are the nuclear membranes. Nuclear membranes. These are the nuclear membranes. Okay, these are the nuclear membranes. So that is nucleus. So nucleus is here. So this one is the nuclear membrane. Okay, that is the nuclear membranes. So in that nuclear membranes, that is, and here what you will have to get? Here that is chromosomes. Here that chromatids. That chromatids here, students, they have been compaction of chromosomes. That is chromatids converted into chromosomes, starting conversion into chromosomes. So that is compact. That is compaction of chromosomes. That is compaction. That is compaction. Compaction of chromosomes. Okay, chromosomes. So chromosomes, and that you see that means here is a cell, animal cell. In that animal cell, that nuclear membrane present. Nuclear membrane present. Okay, nuclear membrane present, and you will have to see the chromosomes, and these chromosomes are of compaction. Compaction of chromosomes. Okay, that compaction of chromosome means that is chromosomes, that is chromatids. Chromatids just starting conversing into that of chromosome. So it will look like that of chromosome. That is compaction of chromosomes, nuclear membranes. And you can have to see the chromosomes in that of that is in ordinary, that is microscope, you can have to see that. Okay, electron microscope is not needed. You can see in that of that is light microscope, you can have to see them. So these are seen, these are visible. Okay, these are visible. Okay, compaction chromosomes, so these are visible. So this is the point that you have to remember, that compaction of chromosome, chromatid look like single thin thread-like structure. That is chromatid, that is thin thread-like structure. This is called the thread-like structure. So this structure generally looks like that of a bucket. Okay, looks like that of a bucket. So bucket stage. So it is also called that of a bucket stage. That is called bucket stage. Okay, that is called bucket stage. Bouquet. So the bucket stage, because these are the thread-like structures. Chromatid looks like these chromatids. Chromatids look thread-like structure. Look thread-like structure. Okay, look thread-like structure. But most important is that of compaction of chromosome. That is important. Compaction of chromosome. Chromatid look thread-like structure. That is called bucket stage, because it is look like that of a bucket. And so that is, and in that phase, that is a nuclear membrane present and visible under, that is light microscope. It is visible under under light microscope. Light microscope. Okay, so that you see that this is that of a leptotin. Leptotin, in leptotin, what happens, that is nuclear membranes present, and that is chromatids look thread-like structure as compaction of chromosomes takes place, and it continues. Compaction of chromosomes starts and will continue. In other phases also, that in next phase also zygotin, that compaction of chromosomes will be shin also. Okay, and the chromatids looks like thread-like structure, and that is the pocket stage. These are the most important events of that of a leptotin. Okay, leptotin. So leptotin nuclear membranes are found visible, that is chromosomes visible under light microscope. Compaction of chromosome and will continue. Chromatid looks like thread structure, and it is as a thread-like structure and look like a bucket. So that it is called the bucket stage. That you have to remember about that of a leptotin. Okay, this much about the leptotin. Very important is that of compaction of chromosome. Chromosome, that chromatid starts converting into that of a chromosomes. Okay, so that compaction of chromosome, it is very important for examination point of view. Bucket stage, chromatid thread-like structure. This much. And nuclear membranes present. Nuclear membrane present. Okay, and that nuclear membrane will disappear at the dikinesis. But in the latter stage of phase 1 in mitosis, that nuclear membrane disappear. Same thing will happen here, but at the dikinesis surface, not here. Here nuclear membrane are found. Okay, that about the leptotin. Okay, students. Then we'll have to move to that of zygotin. Okay, then we'll have to move to that of zygotin. Zygotin. You see that in zygotin, what happens in zygotin? In zygotin, students, you see that is zygotin. Zygotin. In zygotin, it is, students, that is the cell. Here is that of nuclear membranes. Okay, that are also nuclear membranes. That is nuclear membranes. And here in that inside the nuclear membrane, you can have to find out that of chromosomes. So you see that different types of chromosomes. Okay, different size sets of chromosomes are found. You can have to, that is that side. Zygotin also visible under that of that is light microscope. That means you can have to see them like that of leptotin. In zygotin, these are also visible. Okay, visible. You see that. That means suppose you see that. What your students looking here? These are the chromosomes. Okay, these are the chromosomes. You see that. You mark it, students. You mark it that. That means you see that. Which chromosome? Can you say that? I'm just marking it. 1, 2, 3, 4, 5, 6. Can you see that? Which that there are 6 chromosomes here. That means you can identify from that of a whiteboard. That means the diagrams what I have drawn, it might be visible to you. Can you tell me that which two, that means which two chromosomes can form pair? Can you tell, students? You see that one looks like that of 5, one looks like that of 5. So these are the homologous chromosomes. Okay, homologous chromosome. You see that 3 looks like that of 6 and that of 4. So they are homologous chromosomes. Here 3 pairs of homologous chromosomes. You can have to find one, that 1 and then 5. That is homologous chromosomes. Homologous. I'm writing homologous. Homologous chromosomes. Then students 3 and 6. 3 and 6. These are the homologous chromosomes. Again 2 and 4. 2 and 4. They are also homologous chromosomes. Okay, identical and equal shape. That means similar shape chromosomes will have to make pair. Okay, will have to make pair. That pairing, the process of pairing is called that of a synapsis. Okay, that pairing, process of pairing of chromosomes is called synapsis. Okay, synapsis. So synapsis occur. So the process of pairing. So this will have to make pair with that one. This will 3 will make pair with 6 one because they are similar in size, similar in shape. Okay, in looking they are identical. In looking they are similar. Okay, they are of similar shape. Going to that region, they can form what? They can form synapses. So process of pairing of homologous chromosome of homologous chromosomes is called synapsis. That point you have to remember. So the process of synapsis starts in zygote. So there are so many chromosomes. Those are identical. Those are of similar size. Those are of similar shape. They will have to form association. They will have to form pairing. So these are called the homologous chromosomes. Those chromosomes look alike. They are called homologous chromosomes. So these chromosomes, so one will pair with 5, so that will synapsis will made. So 3 will pair with 6, synapsis will be made. 2 will pair with 4, synapsis, that is synapsis will be made. So there will be more, that is synapsis will have to find in that of pairing. Okay, so that possible is only in that of homologous chromosomes. And you see that means pairing. Suppose one is, you see that this pair with that of this one, this pair with that, they are similar, they are homologous chromosomes, so they will have to make pair by which means they will have to, you see that. Suppose one student says it is dearest, nearest and dearest to that of another friend. It is because of that of some bond, not peptide bond, but that is bond of belief. Okay, bond of belief, bond of faith, bond of faith. That is bringing two friends together. Okay, so similarly, that means there is certain chemical, there is a certain protein. That protein brings these two homologous chromosome together. Okay, brings that homologous chromosome together. And what are these proteins? You'll have to know that is called Synaptonomium Complex. Okay, that is called. You see that I'm just drawing it nearing to it. Okay, you see that this one and this one. That means this will make pair with that one. And there is a protein, there is a chemical which can have to bring these two chromosomes together. Okay, pairing is possible only to that of presence of that of a synaptonemal complex. That is synaptonemal complex. You see that this one. This one is a synaptonemal complex. Okay, synaptonemal complex. That is called synaptonemal complex. Okay, synaptonemal complex. This synaptonemal complex, it is a protein, it is a chemical, it is a proteinous structure, it is a protein parts which bring two chromosomes together. Okay, which brings two chromosomes together. That means two chromosomes can be attached to each other, can make pair. They are here and here. That means one is here, another one there is a, so how they will have to come together? They will have to come together and make a pair by means of a protein, by means of a chemical. That chemical made up of protein, that is called synaptonemal complex. Okay, synaptonemal complex will bring two chromosomes together. Okay, so this is a protein part. And so the process of pairing of homologous chromosome is called, that is synapsis, and that protein which requires for that of a pairing is called that of a synaptonemal complex. Okay, that point you have to remember. Then you students, you mark that of then there is a bivalent or a tetrad. You see that. That means you see that. That means this is a one, another one I am drawing here. Okay, you see that. That means there is a protein, that synaptonemal complex. Okay, that make these two together. So these two will form a bivalent. These two will form a bivalent. That is called bivalent. Okay, bivalent. These two will form a bivalent. Bivalent in the sense means there are two chromosomes. There are two chromosomes. This is chromosome number 1. This is chromosome number 2. So two homologous chromosomes after pairing will make that of a bivalent. Okay, that bivalent means that is two chromosomes, but it is also called tetrad. It is also called tetrad. Why it is called tetrad? Because there are four chromatids. Because there are four chromatids. You mark it that one chromatid, two chromatid. Here is that three chromatid, four chromatid. So this bivalent is also called tetrad. If you will have to count the number of chromosomes, chromosome number is 2, so they are called bivalent. Such pairing of homologous chromosomes are also called tetrad. Why they are called tetrad? Because there are four chromatids meeting together, four chromatid pairing together, so they are called tetrad. Okay, so the main point in zygote is that compaction process continues. That is the process of them that same look like similar looking chromosomes will are called that of homologous chromosome. Such homologous chromosome will start pairing. Those are similar in shape, similar in looking. They will have to make pairing, and that pairing is called synapsis. And that synapsis is possible due to a chemical made up of protein that is called synaptonomial complex that will make the two chromosomes together, and that pairing is called bivalent. Bivalent means that is two chromosomes, and such bivalent is also called tetrad. Why it is called tetrad? Because there are four chromatids that unite together in a bond, okay, in a pair. So that is called that of four. These are the four chromatids. Owing to chromatid, tetrad. Owing to that of chromosome, bivalent. This is about that of a zygote. Okay, students. So zygote complete. Then we'll have to come to that of a patching. Okay, we'll have to come to that of a patching. You see that in patching, that is those are tetrads. Tetrad are somewhere visible. Tetrads are formed in zygote. You make that is a concept clear that tetrads are formed in zygote, but tetrads are visible. What can you tell me that? That means such tetrads or bivalents. Tetrad or bivalents are formed in which surface, students? In which surface?
Zygote.
Zygote, but they are clearly visible in the subphase of Pachitin. Next page is Pachitin, where they will clearly visible. Okay, that means that they will be clearly visible. Tetrads will be clearly visible. Bivalent will be clearly visible in that of subphase of Pachitin. But generally, they are formed in the subphase of zygote. Okay, now we'll have to discuss about that of Pachitin. Okay, and Pachitin, you know that means leptotin and zygotin, they are that of for short duration. But Pachitin is it is a longest subphase. It is the longest subphase of that of all five phases. Okay, subphases. So it is the longest one of five subphases that you will have to remember. Which one is the longest subphase in that is prophase one? Answer will be that is Pachitin. Okay, that is Pachitin. You see that Pachitin. In Pachitin, that is nuclear membrane also present. Nuclear membrane present. Nuclear membrane present in leptotin, in zygotin, in Pachitin, in that of diplotin. But nuclear membrane will disappear in the dikinesis. So nuclear membrane are present. I am not drawing it again, that you know that of drawing. So no need. I am just showing you. Okay, you see that, students. I am drawing that two. Okay, that two. I am drawing here. You see that. I am drawing here. You see that. That means you see that. That means this is a chromosome one. This is a chromosome two, and these are you see that. You see that. What I'm just telling you that before I'm just telling. After that I will draw. Okay. You see that. You see that. This is chromosome one. This is chromosome two. So this is you mark it A and B. In what is the relationship between A and B? that A and B are sister chromatids. Are sister chromatids. Okay, are sister chromatids. Here also C and D also that of sister chromatids. Okay, they are also sister chromatids. Okay, sister chromatid. You see that A, B are sister chromatid of chromosome 1. C and D also sister chromatid. Okay, but if you have to come relations of B and C will be sister. They will be that of a non-sisters. Okay, non-sisters. They are non-sister. B and C are non-sisters. Okay, they are non-sisters. But here, that means here you see that. Here you see that genetic material, that is genetic material will be transferred from that of chromatid number C to chromatid number B. Okay, in from that of that is called crossing over. So crossing over in Tacitin, main thing occur that is called crossing over. Okay, CO. Crossing over takes place in Tacitin in between two non-sister chromatids of homologous chromosomes. You see that this is a homologous chromosomes. You see that A1 and 2 are these are 1 and 2 are homologous chromosomes. They are identical in looking, similar in looking. So they are what? They are homologous chromosomes. And in homologous chromosome of 1, A and B are two sister chromatids. A and B are two sister chromatids. Similarly, for that of chromosome number 2, that C and D are sister chromatids. But if you have to compare between 1 and 2, one chromosome number 1's chromatid B with that of chromosome number 2's chromatid C, then what relationship we'll have to find? That means relationship is that they are non-sister. Okay, so crossing over is the process by which genetic material transfer in between two non-sister chromatids of homologous chromosome. So crossing over is a process of transfer of genetic material in between two non-sister chromatids of homologous chromosome. Two non-sisters. So crossing over is a process of transfer of genetic material in between two non-sister chromatids of homologous chromosomes. Okay, so what is crossing over? Crossing over is a process of transfer of genetic material in between two non-sister chromatids of homologous chromosomes. These are homologous chromosomes, but that genetic material will be transferred in between B and C. So you see that I'm drawing so that in that process you see that you see, you mark it that means here that is chromatid comes and bend with that of, so make a wrapping, so that wrapped with that of another, so that is called crossing over. Okay, that is called crossing over, and this point you might mark that. Okay, this point you mark that is called recombinant nodule. This point, this point, where one is mixed with another, joins with another, one chromatid joins with another chromatid or wraps with another chromatid at the points. The points at which a chromatids wrap with another chromatids is called, that is recombinant nodules. Okay, that is called recombinant nodules. What it is called, students? That is called, okay, that is called recombinant nodules. Okay, recombinant nodules. That is called recombination. That is called recombination nodules. Okay, so crossing over takes place at which site? Answer will be recombination nodules. Okay, recombination nodules. And that is also crossing over is also students enzymatic process. Okay, enzymatic process. The enzymes which help in that of crossing over is called that recombinase. Recombinase. Okay, which enzyme? Which enzyme gives support for that of crossing over? Answer will be recombinase. Answer will be recombinase. Another, that means in your NCERT book that recombinase has been given. But another two enzymes also required for this purpose. One is that of endonuclease. Another one is ligase. Okay, there are two other enzymes also that are required for that of a crushing over. They are called endonuclease ligase. So moreover, three enzymes are responsible for that of wrapping of that of chromatids with one another, or that of crushing over. That crushing over is possible due to that of three enzymes. They are called recombinase, endonuclease, and a ligase. That you will have to remember. So recombination nodule is a site where crossing over takes place. It is an enzymatic, that is enzyme related. So it is an enzyme related process, whereas recombinase, endonuclease, and ligase that help in that of crushing over. And the site where crushing over takes place is called that of recombination nodules. Okay, recombination nodules. Then students will have to find it that is exchange of genetic material. So what is the objective of that? What is the objective of crossing over? Is the transfer of genetic material. Okay, transfer of genetic material from one chromatids of a chromosome to another chromatid, non-sister chromatid of another chromosome. So transfer of genetic material is the most important objective of that of crossing over. Okay, crossing over. So that is called crossing over, and it happens in that of recombination nodules. Recombinase, endonuclease, and ligase are helpful for that carrying out such process. Okay, then we'll have to know about that of diplotin. Okay, next is that of diplotin. Diplotin, you see that in diplotin you mark that. In diplotin, what happens, students? You see that in diplotin you can have to see. That means here is that there is, you see that it is a one chromosomes, it is another chromosomes. Okay, you see that this chromosome and that chromosomes. That means these chromosomes chromatid, another chromosomes chromatid, just make a except structure. That structure is called Kaismata. Okay, so Kaismata. So X-like structure is found in that of diplotin. In that of diplotin, you can have to find out that of X-like structure. This is a X-like structure, and this is called, students, Kaismata. Okay, that is called Kaismata. Kaismata. It is a except structure. Except structure. That means you see that this one is that of what? This one is that of a chromosome. Chromatids of a chromosome. This is also chromatids of chromosomes. One chromatid, that means one chromatid meets with that of another chromatids, and it looks like that of a except structure. And this except structure is called Kaismata. So Kaismata is found in that of diplotin stage. Okay, this is a except structure, that is Kaismata. And Kaismata is formed when that two chromatids of opposite chromosomes make that structure. Okay, make that structure. So this is a chromatid, this is another chromatid. They will make like this structure. So this is called Kaismata or except structure. Okay, except structure. And in that also, in that of diplotin, that dissolution of synaptonomial complex. So synaptonomial complex that are found that protein, they will start disappearing. That means it will start disappearing. That means it will seem no more. So dissolution of So dissolution of that is Synaptomal complex. Okay, Synaptomal complex. So it will not sing anymore. So Kaismata except structure are found. Okay, another thing is that. Okay, another thing is that. That means you see that in women generally you see that when a girl child that is takes birth, before her birth, that means the process of oogenesis. The process of oogenesis. Okay, that is in a female, that is a girl child, in a girl child before her birth. That means at that of when it is at embryonic stage, when girl child is in embryonic stage, at that time oogenesis starts. Oogenesis starts means that is formation of ova. Okay, formation of ova. You will be surprised that there are some that is large amount of that is ova are present in that of that is female child, and that process of producing ova continues even that of till that of 13 to 14 years of age when they will have to get the maturation. So their ova remains at that of a diplotin stage. Okay. After that getting maturation, that is a girl generally gets matured at the age of 13 to 14. So that after such maturation, that means a diplotin arrests ova diplotin stage arrest ova, that is production of ova till the age of 13 to 14. After that, the production of ova will start as menstrual cycle will start in the female. So diplotin stage, what it is does, that is arrest the release of ova in that of a female child up to the age of 13 and 14. Okay, so this is the dissolution of synaptonomial complex, formation of Kaismata except structure, and that arrest of ova in that of a girl child up to the age of 13 to 14 is found in that of a diplotin stage. Okay, then we'll have to know about the last subphase, then we'll have to finish. Okay, that is dikinesis. Dikinesis. In the dikinesis, you see that is the dikinesis stage. That is dikinesis. That is dikinesis. Okay, dikinesis. In dikinesis, you see that in that cell, you can have to no longer found that of nuclear membrane. Okay, nuclear membrane are absent. Nuclear membrane are absent. So nuclear membranes are absent. Okay, we'll have to find the no nuclear membranes. Okay, no nuclear membranes, and that chromosomes. Okay, about that chromosomes are fully condensed. So the chromosomes are fully condensed. Condensation. Chromosomes are fully condensed. That is chromosomes are fully condensed. Okay, condensed. That is what in that of a prophase of that of mitosis, that is condensation of chromosome. Same thing students happen, chromosomes are fully condensed at that of a dikinesis, and then nuclear membranes are absent. Nuclear membrane are not found. Similarly that of a endoplasmic reticulum, similarly that of a endoplasmic reticulums not found, nucleolus not found, Golgi bodies are not found. Similarly, they are also absent. Okay, they are also absent. And in that phase also, nucleolus disappear, nuclear envelope breakdown. Then this one meiotic spindle assembly. Then that is meiotic spindle. That is meiotic spindle. So spindle formation start in that of metaphase. You can see that how the spindle fiber, okay, take that of chromosomes, and so that meiotic spindle formation starts. Okay, formation starts. So this is the important events that of dikinesis and transition to metaphase. And that dikinesis will transition, that is transition to metaphase. Okay, that transition to metaphase. So in that phase, what you have to find out that is chromosomes are fully condensed. That means that chromosomes are fully condensed. So these are the chromosomes that are fully condensed chromosomes. So that is, you have to remember, nuclear membranes absent, chromosomes are fully condensed, transition to metaphase phase, meiotic spindle formation starts. Okay, so that is division will occur, so that meiotic spindle formation starts. So this about that of prophase one of that of meiosis one. I have already told you that of 5 phases. Okay, if you have doubt, you can have to ask me also, and you have to contact with me on that of WhatsApp app also. You can have to contact with me if you have doubt in your mind. Okay, students. So tomorrow we'll have to discuss about that of interkinesis. Then we'll have to move to that of meiosis 2. And meiosis 2 will have to complete, that is same to that of mitosis. Then we'll have to significance of that of meiosis. After that, we will have to complete this chapter. From day after tomorrow, we'll have to start a new chapter. I have decided to start with that of animal physiology. Okay, that 7th chapter will have to take 28 days. Then in February months, you will have to finish that of that chapter. So the 7th chapter of that of human physiology. After that, in month of March, you will have to complete that of 5 chapters of plant physiology. By this process, you will have to complete all the course what CBSE has given to us. Okay, so that in this way today I am concluding, and I can't ask any question because another class is there, so that you should have to appear that class, attend that class. So I'm concluding here. Thank you, students. Thank you.
Thank you, sir.
Thank you,
Thank
sir.
you, sir.
Thank you.
Thank you, sir.
Thank you.
Thank you, sir.
Thank you. Thank you. I have not that one. I'm just doing that one.